student who show it in the graph file. So, for example, Phạm Đức Gia Hiển, Nguyễn Ngọc Nhi, Đỗ Trọng Đạt. So you are not put in the file in the draft state, in the submitted status. You put it in the draft status. And when you put it in the draft, I cannot put a grade on your file. Okay, so you have to put it in the submitted file. And I set the deadline, I reset the deadline in the end of today. That means you have roughly more than 15 hours more to submit your file. So if you have not submitted your file, please remember to submit it. 15 hours, okay, for the last submissions. So now we will come back to chapter 4 in the genetic variation and marker. Just to remind for the newcomer that you only have 15 more hours to submit your file, your bioinformatics practical assignment, okay? Alright, so talking about genetic variations. Most of the people, humankind, is the same with each other in 99.9% of the genomes. We are only different with each other in 0.1%. That means both people in African, people in the Arctic, people in Asian, people in Africa, in European, are the same in 99.9% of the genomes. Only 0.1% are different. The human genome is roughly about 3.2 billion bases. That means that we are only different with each other in around 3.2 million bases. But this over 1% are determined that they determine differences in our physical trait. Some people are born with dark hair. Some people are born with blonde hair. Some people born with red hair. The eye color, the blood type, the other physical traits like height, weight, freckle in the skin, or the shape of your hair is whether is straight or wrinkle. So these genetic variations also determine the difference in the susceptibility to a certain type of disease. For example, some people are more susceptible with cancer. Meanwhile, the other group of people are quite resistant to several types of cancer. Even they have all the lifestyle risks. For example, they smoke, they drink, they not having a healthy diet, but they still have a lower risk to cancer compared to the others. Genetic variation also identify the different response to certain medications. So we have discussed about the codeine, which is the substances to treat with nervous, and it's calm down the mind, and inside the normal people, it will transfer into morphine to calm down the mind with a slow rate. But in certain cases, for example, one mother in US, where he using codeine because he's have a genetic factor that transforms the codeine too fast, leading to the morphine concentration in their blood is too high. That kills the baby through a breast milk. Okay. So, from this, genetic variations will determine different features in our both physical and mechanisms in particle. And study about this genetic variation leading to now we have a 10,000 genome program. In 2018 we have a 1,000 human genome to identify what is the main differences in human kind. And now we even have 10 countries have reached the 100,000 genomes. That means they have sequenced more than 100,000 of their citizens. In Vietnam, where 1,000 people have participated in this project to identify what is the main differences in the genetic background of the Vietnamese compared to the others.
Humans around the world have much in common, but also enormous diversity. Some of the differences between each of us come from our environment and life experiences, but our DNA plays an important role in determining our appearance, our traits, and our health. There are thousands of genes in the human genome. Sequence changes in individual genes can determine if we have freckles, can digest lactose, have wet or dry earwax, are red-green colorblind, or are likely to have blue eyes, or think broccoli tastes better. Individual genes can also determine if we will develop sickle cell anemia, cystic fibrosis, or Huntington's disease. Multiple genes act together with our environment to determine our hair and skin color, our height, our weight, our blood pressure, and our risk of developing type 2 diabetes, depression, cancer, some autoimmune disorders, and many other conditions. In spite of all these potential differences, humans are 99.9% genetically identical. How is it possible that we are all so similar and yet so different? Let's zoom in to the smallest genetic unit, a single nucleotide of deoxyribonucleic acid, or DNA. A DNA nucleotide is composed of sugar and phosphate groups, and one of 4 nitrogenous bases: adenine, thymine, guanine, and cytosine, annotated in shorthand as A, T, G, and C. The sugar and phosphate groups form the DNA structural boundary, allowing nucleotides to concatenate into a long single strand of DNA, while the bases determine DNA sequence. The chemical properties of DNA allow bonds to form between the bases in order to create a double strand, with 2 hydrogen bonds pairing A and T, and 3 pairing C and G. Though different types of human cells can be very different in appearance and function, they contain the same genome, which consists of about 3 gigabases, or 3 billion base pairs, of DNA. All the DNA in the cell would be about 2 meters in length if it were stretched out, and must be condensed down to fit into cells as small as 10 micrometers across. The DNA is first coiled into its canonical helix structure, and then wrapped around histone proteins to form a DNA protein structure called a nucleosome. These nucleosomes can be further wound and coiled together to create a compact structure that fits into the nucleus. During cell division, the DNA is organized into tightly wound chromosomes, 46 in total, with 23 coming from each parent. These chromosomes can be easily and accurately separated during cell division guaranteeing that each new cell contains an exact copy of DNA. Outside of cell division, the DNA is decondensed in the nucleus, allowing greater accessibility. The transcriptional machinery regulates expression of the approximately 20,000 genes, which, although they correspond to less than 2% of all genomic DNA, encode all the proteins necessary to build and run a human cell. So to return to the original question, how is it possible for all of human diversity to exist when we are 99.9% genetically similar? It is important to remember that the 0.1% of DNA that varies, on average, between each of us, actually corresponds to about 3 million differences across the genome, with 20,000 of them, on average, falling into protein-coding genes. Although that equals approximately 1 difference per gene, in reality, these differences are not evenly distributed across the coding region. Differences in DNA sequence are called variants, and those affecting a single position are called single nucleotide variants, or SNVs. Common SNVs that occur in more than 1% of the population are called single nucleotide polymorphisms, or SNPs. SNPs, along with larger scale sequence changes like deletions, duplications, and rearrangements, create all the richness of human genetic diversity at the population level. This raises many questions on an individual level as well. How does DNA determine our traits? How can we understand what a gene does, and how variants in that gene might affect our lives? What can our DNA tell us about our risk and our loved ones' risk of disease? How can information from our genomes improve our medical care? Understanding genetics allows us to apply the concepts of heritability and genetic variation to questions of human health and disease in our world today.
So, studying genetic variation and how it affects the genetic variation affect to our physical trait can lead to understanding on the function of genes and what is the role of gene do. What we call now genome annotations. Genome annotation is to annotate the genome to know which is function of each fragment of genes. Some genetic variations identify differences in physical traits. Some traits are quite normal, but sometimes the mutations can lead into quite extraordinary traits. For example, you are looking at Stephen Crump, or we call the man who cannot catch HIV. So HIV is a lethal disease. When the virus infects into the body, it will attack into the T4 lymphocyte cell. And with normal people, the HIV cannot be cured. We have now the drug to low down the rate and prevent the virus can multiply with the high ratio, but we cannot cure HIV or AIDS. But in the case of Stephen Crump, this man was born with an extraordinary innate capability that his body cannot get infected by HIV. So it comes from a phenomenon that Stephen actually is homosexual guy. That means he fell in love with the other guy. So meanwhile, his partner was regularly killed by HIV. He seemed to be healthy and unaffected by the disorder. So when the scientists scanned his genome to see what made it has the ability to prevent HIV, they find out mutations. Delta 32 mutation. in the CCR5 receptor. So this receptor was play a role in the membrane of the CD4 cell. With normal people, the HIV will infect the CD4 cell through the receptor in the membrane of the CD4 cell. However, in case of Stephen Cron, because he had a mutation of delta-32, delta is a short synonym for deletion. That means he had a deletion in the amino acid 32. Okay? And this mutation leading to, because this protein is a structural protein, so it leading to the modify in the structure of the CCR5 genes, CCR5 receptor. This modified structure are not affected into the function of the CD4 cell, but it leads to ability that the HIV cannot recognize this is a cell it has to infect it to. So Stephen was protected against HIV. So thanks to this significant discovery, nowadays scientists have developed a drug called Maraviroc. Maraviroc so this drug will block the CCR5 receptor so that prevent the HIV virus infect into the carrier. One funny thing about Stephen Cron is his grandfather is Dr. Cron, who was also a medical doctor and who discovered about the bowel Crohn disorder. So, thanks to the case of Crohn and thanks to study his extraordinary genetic background, scientists now have invented the Maraviroc to treat the HIV patients. And recently, in 2018, I guess that you all heard about this story. In the end of 2018, we have a scientist, actually he's physics. He studied in biology and he's learned about the technology called CRISPR. We'll discuss more about this powerful tool in tomorrow. But this tool helps scientists to edit in the genome to transfer a mutation at the position of interest. So, He Zhang-Kui, Chinese scientist, based on the case of Stephen Cron, who innate have a delta-32 mutation naturally, but in this case here, he had created two twins baby girls based on CRISPR-Cas9 and create artificially delta-32 mutation. And this created two baby girls, and he claimed that he did that to protect the two baby girls against the infectious risk from the father who gets HIV. Let's hear that what her claim about this study.
You may wonder why we use the gene surgery to prevent HIV. Why not another method disease? Two reasons. First, safety. Second, real-world medical value. As a scientist and the father of 2 girls, safety is my #1 concern above all else. The gene we choose, CCR5, is one of the best studied genes. In fact, about 100 million people naturally has genetic variations that disable CCR5, protecting them against HIV. Live a healthy. Our surgery caught exactly at the start of the natural variation. Decades of clinical trials has tested medicine mimicked that natural variation, including the first genetic trial in the United States. Well, it proved safe. Also, protecting against HIV requires the simplest gene surgery, which is to remove just a few DNA letters. Correcting a broken gene will helpful for more diseases like familial cancer or muscular dystrophy. It's more complex. Studies in the US haven't shown this works well in embryos yet. The second reason we end up choosing HIV as our first target is the real-world medical value. HIV remains a deadly and devastating infection that needs medical advances. About 1 million people died of HIV last year. New infections remain 3 times higher than the UNAIDS target for 2020. No vaccine or cure exists. Taking pills daily from night carry healthy risks. In many countries, discrimination deepens the devastation of infection. Employer might fire people after discovering their HIV status. Doctor may refuse to treat CCR5 surgery. While not applicable to the general public, it may be valuable to help a few affected and very high-risk families protect their children from the same fate. This leads to the National Academics in 2017 named vaccine-like disease prevention as an ethical application of gene editing.
So the purpose of her is good, but until now, even in Chinese or around the world, genome editing human beings still be banned due to certain ethical issues. So as the information I know, after published his shocking research on the 2 baby girls, He was fired from his university because he's performing the lethal experiment on a human subject, and later was putting in jail by the same reasons. So let's talking about genetic variation and marker. What is the genetic marker? Genetic marker is differences in the genetic background of a human being compared to others. That is differences between individuals and individuals. There is many types of genetic marker, but in our training program, due to the limitation of the time, I will only introduce you 3 types of marker, that is the VMCR, SNP, and SFR. Okay? So first talking about the STR, which stands for short standard repeat. So short standard repeat is a repetition of a fragment from 2 to 4 nucleotides. For example, in here we have CTT, 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 which repeats 6 times. In another people, we have this CTT repeat 1, 2, 3, 4, 5, 6, 7, 8 times. So based on electrophoresis, that we put it in negative here and positive here. Remember that DNA in the normal pH equal to 7, will have a negative charge, right? Because we have a phosphor, because DNA is polymerization nucleotide. Nucleotide is composed by 3 main parts, a phosphor group, 5 carbon sugar, and base unit. And the phosphor group is a negative charge at neutral pH. So DNA is normally negative charge at the normal pH, and if we have put in here electric currency, we'll see that it will move from a negative charge to positive charge. The distance it moves will be the same this side. Another marker is called VNTR. VNTR is a variable number. So we have 2 types. The SDR, which stands for stop, tandem, repeat, is repetitions from 2 to 4 nucleotides. And the VNTR, which stands for variable number time repeat. Short time repeat and variable number time repeat is the repetitions of the fragment more than 4 nucleotides. Okay? So for example in here we have a repetitions of a fragment composed by 33 nucleotides. So in person A, we have the first one is 4 repeat, and the second allele is 6 repeat. This marker is also repeat on another locus. We have 3 repeat, and the other allele is 7 repeat. In another person's, in the first locus, we have 2 alleles. We have 2 repetition and 3 repetition, and the second locus, we have 5 and 4 repetitions. So when we have run electrophoresis from a negative charge to a positive charge, we will have a profile of person A and B here. We have 7, 6, 4, and 2 repetition. 7, which corresponding for this fragment. 6, corresponding to this fragment. 4 and 3. And the person B will have 2, 3, 4, and 5. So 2, 3, 4, and 5. The smaller fragment is, the faster it will move in the electrophoresis. So let's talking a little bit about technical issues here. How we can amplify the SDR and VNTR marker is based on the technique that was invented in 1981 by Lewis. We call PCR, or polymer chain reaction. Now they put PCR is a fundamental experiment that you can perform in any molecular biology lab. So this is a technique that helps us to amplify a fragment DNA of interest. For example, you want to amplify a gene that coding for the CCR5 receptor, or you want to amplify a gene that encoding for insulin in bacteria. you can amplify the fragment by PCR technique. So, in PCR, what is PCR? PCR in a term of technical issues is a technique using to amplify a fragment based on the same principle that with the DNA replication in natural. First, we must denaturate the two DNA fragments. So in DNA replication, we do it by they do it by the DNA helicase, right? The DNA helicase is tear down the hydrogen bond that links two DNA strands together. But in polymers, PCR, we do it by temperature. Just put it in the temperature of 94 to 96 degrees C, and it is better. Later, there will be a primer. Also in DNA replication, we have primer. The primer in there is RNA, but in there, primer is DNA. And we have a DNA polymerase to amplify the fragment. So, after the temperature was going to 3 steps here, it will return to the beginning. And from one DNA strand, after one cycle, we will have 2 DNA strands. After another cycle, we will have 4 DNA strands. And after another cycle, we will have 8 DNA strands. So after each cycle, we will have the number of DNA strands was doubled. So, all the playing role participate in PCR. And to perform PCR, we need to put it into the machine called a PCR machine or a thermocycle, which will turn the temperature from denaturing at 65 degrees C, annealing with the temperature corresponding to primer, and expansion is temperature nearly to 72 degrees.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the prime sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
So after each cycle, the number of DNA strands was doubled and doubled until the fragment in the beginning was replicated millions or even billions of times. So that's the first step, to amplify the gene of DNA rest. And second step, we will have put it in the electrical current series to separate the different fragment here. Okay? So we perform it by a technique called gel electrophoresis. So gel electrophoresis, that means just put the DNA fragment into a currency so it will scrape each other, and based on the distance it's moved, we can calculate what is the size, what is the average size of the DNA fragment. So it's based on the moving in the currency of the DNA fragment. So the DNA fragment here, you put it here, and this is the gel that made by agarose. Agarose is the same component with the other that sometimes your mom or your grandmother made for you at home, especially in the summer holiday. But for agarose, it must be ultra cool. Okay? So the DNA put it in here, and when we put it in the negative charge and positive charge, because DNA in the neutral pH is negative charge, so it will be moving far away from a negative charge to the positive charge, move away from the cathode to the anode. Okay? And based on the distance it's moving in the period of time, we can calculate what is the average size of the DNA fragment. After the electrophoresis have finished, we'll use the atomic bombard. You remember these substances? So I have mentioned about these substances once when we're talking about mutations. These substances is one of the highest substances that can cause DNA mutations. Okay? But it also has an effect that it will bind in a DNA double strand, and it will provide a light deflection in the UV light, the light that have the wavelength less than 400 nanometers. Based on the light of the DNA fragments, we can identify what is size of the DNA fragment.
Once DNA has been extracted from a sample and a section bearing useful code such as a VNTR or STR is isolated, the next step in DNA fingerprinting is to measure each strand and count the number of repeated sections. To do this, scientists use a technique called gel electrophoresis, which uses an electric current to push strands of DNA through a slab of gel-like material. Because each bit of DNA is negatively charged and subject to an equal electric force propelling it to the positively charged side of the gel, smaller pieces move faster than larger ones. When the current is removed, the gel is photographed to reveal how far each bit has migrated. By comparing the bands produced by the DNA samples of interest with bands produced by standard samples whose sizes are already known, the precise length of each DNA fragment can be gauged.
So based on this technique, people can perform a profile of the DNA marker. Microsatellite marker is composed of STR and VNTR. Microsatellite marker. Is composed by both VNTR and STR. So for example, we have a person here. We have a marker. It can be STR, it can be VNTR, whatever. It's a repetition marker. So in here, First, we will amplify the fragment using PCR, the technique that we just discussed. And then we run it in the electrophoresis drill. For example, the couple here having 6 offspring, okay? And she has contained mutations, okay? Here contain mutations in the 3, in the number 2 here, which are. So this offspring, this couple have 6 offsprings, and as you can see, each of the offspring receive 1 allele from the mother and the other allele from the father, right? So for example, the mother has 2 alleles, one with 3 repeatations, and the other with 5 repeatations. When his profile will run in the electrophoresis, we have the number 3 here and number 5 here. Number 3 is here, it's moving faster because it's smaller, and the number 5 is here. Okay? The father has 2 markers. The number 2, that means 2 repetition, and 8, that means 8 repetitions. The H is the longest, so it will move the most slowly. And the 2 was most fast, so it's moving forward. So this is the DNA profile of father and mother. Now, the first offspring, he received the first, that means the 5 from the mother, and the second one is number 2 from the father. So the profile of the number 1 is 2 and 5. 5 from the mother and 2 from the father. The second offspring will inherit the 2 biggest ones, the 5 from the mother and the 8 from the father, so 5 and 8. Now the third offspring, he inherited a marker, the 8 from the father and the 3 from the mother here. So 3 and 8. And so on for the 3 other child. So now I will have a question for you. We have been through the exercise tutorial. We know about the chapter number 3 when we're dealing with Mendelian inheritance disease, right? So now suppose that this family suffer from a disorder. The mother is a carrier of the disorder, and in 6 offspring, 4 will carry the disorder. Please tell me that the disorder is dominant or recessive, and 2, in the 2 allele, in the 4 allele here, which allele was close related to the disorder? You understand the question, right? Or you not understand the questions? No. Okay. Re-explain it in Japanese. Ở đây chúng ta có một cái phân tích kiểu gen microcellular marker, nghĩa là cái chỉ thị vệ tinh DNA của một cặp đôi bố mẹ, và bố mẹ này có 6 đứa con, chưa? Ở người mẹ thì có 2 allele, chỉ thị thứ nhất là lặp lại 3 lần, chỉ thị thứ hai là lặp lại 5 lần, ok chưa? Và cái 3 lần thì là vì là nó nhanh nhẹ nhất, cho nên là cái 3 lần là nó đang ở đây. Cái 5 lần là nó nặng nhất, nó đang ở đây. Thì trong quá trình điện gì thì cái nào càng nhẹ thì chạy càng nhanh, đúng không? DNA tiếp điện âm, cho nên khi đặt vào hiện trường thì nó sẽ chạy ra khỏi đường âm và chạy đến đường dương. Đoạn DNA càng nhẹ thì nó chạy càng nhanh, được chưa? Thế thì đây đoạn chạy càng nhanh, chạy từ đầu này đến đầu này, đoạn chạy nhanh nhất chính là đoạn lọc lại số 3, đoạn chậm nhất là đoạn lọc lại số 5. Chưa? Người mẹ là người mang bệnh. Bố là có cũng một cái chỉ thị vệ tinh nữa, lặp lại 2 lần và đây là 8 lần. Như vậy đoạn 2 là số 2 ở đây và đoạn số 8 ở đây. Được chưa? Và đây là profiling của 6 đứa con. Mỗi đứa thì sẽ nhận một alen từ bố và một alen từ mẹ. Được chưa? Ví dụ đứa này thì nhận alen thứ nhất là 3 đoạn từ mẹ và alen thứ hai là 8 đoạn từ bố. Rồi. Đứa thứ hai thì sẽ nhận là 3 đoạn. Cái này là 8 từ bố và cái này là 5 từ mẹ. Đúng không? Đứa thứ ba thì nhận 8 từ bố và nhận là 3 từ mẹ. Và tất cả những đứa còn lại. Bây giờ người mẹ có một căn bệnh di truyền cho 4 đứa con. Và cái gen gây nên căn bệnh đó sẽ nằm ở chromosome này hoặc chromosome này. Bây giờ câu hỏi thứ nhất là căn bệnh này là dạng cho bé dạng nặng. Đừng trả lời chắc chắn thế. Và câu hỏi thứ hai là dựa vào cái gen profile này, hãy cho biết là cái gen gây nên căn bệnh đấy là nằm ở đâu trong 4 chất thể. Ở đây chúng ta biết là từ mẹ rồi đúng không? Thế thì nằm ở những chất thể nào của người mẹ trong hai những chất thể này? Có hiểu không? Hiểu câu hỏi không?
Xác định cái căn bệnh này nó nằm ở cái
Đúng rồi, nó nằm ở cái alen trên hay alen dưới.
Thì thầy trên nói tiếng Việt ạ. Thì idea của em là em nghĩ là nó là cái trên ạ. Tại vì khi mà vâng là cái đấy, vì như thầy có giải thích thì là nếu như mà nó chạy nhanh hơn ấy, thì nó sẽ ở trên, thì em nghĩ là nếu mà nó chạy nhanh nó sẽ phát bệnh hơn.
Có ai có đáp án khác?
I think it's the second one because the 1, 2 characteristics chart, whenever the second one goes with the number 1 or 4, it always will get like that. Thì nó sẽ thuộc về
So that is the correct answer. So we can see here, whenever the offspring is come with the 5 marker here, 1, 2, and 5, 6 have the 5 marker from the mother. It always having the disease, right? The 1, 2, and 5, 6 is inherited the 5 from the mother. Only 3, 4 is inherited the 3 from the mother. And 1, 2, and 5, 6 carry the disease, not the 3 and 4. So that is linkage. So in here we can observe both Mendelian inheritance and linkage inheritance. Mendelian that related to the microcellular marker, and linkage that each marker will have a tie linkage. The marker number 5 here will have a tie linkage with a genetic causing the disorder. All right. So based on the VNTR and the STR marker, we can also provide a profile of the DNA fingerprint. So for example, in here, I have 3 markers, BNTR, BNTR 1, 2, and 3. And analyze in 3 different individuals, A, B, and C. We'll see that in the first individual A, we will have 3 repetition and 8 repetition. 3 here and 8 here. The second locus is 3 and 6 here, and the other one. So come along with 3 markers and build up a DNA profile of the 3 markers in this individual. And based on this, in 1970s, Jeffrey, a scientist, US scientist, had invented DNA fingerprinting based on different markers. So, what is application of DNA marker? First, it will be applied in pattern testing, like the test we saw. Father and mother have 3 children, and each child will inherit at least we inherited 1 allele from the mother and 1 from the father. For example, in this case, the father has 11 repetition and 14 repetition of SPR marker. Mother had 8 and 12. And so the child will have 12 and 14, 12 from the mother and 14 from the father. Second child will have 8 and 14, 8 from the mother and 14 from the father. So, based on this, the FBI had published a 16 SPR marker that using for paternity testing and chemo investigation. And this is locations of the 16 SPR marker. So in chromosome number 1, we have none. Chromosome number 2, we have 2 markers. 1 in the short arm and another one in the long arm of the chromosomes. Number 3 we have 1 and so on. In X we have 1 marker. 16 HPR marker. 1, 2, 3, 4, 6, 7, 5, 6, sorry. 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. 16 HPR markers. Published by HPR. And this was applied for many tests. For example, we have the case here for paternity test with the DNA profile of mother, child, and 3 suspect deaths. So we have a case here, even the mother can tell who is the real father of the child. Based on the DNA profile, could you help me to identify who is the father of this child? Number 1? Alright, so we have the first solution is number 1. We have another solution. Now, what is solutions? 3. 2. Okay. Who think is the first one? Please raise your hand. Okay. Who think is number 2? Please raise your hand. We have 3, 4, 5, 6, 7. Okay. All right. A lot of people think it's